Okay, good afternoon, everyone. Welcome to our Breakthroughs in Cancer seminar series from the Stanford Cancer Institute. Welcome everyone who is here in person and everyone joining on Zoom. It's my pleasure to welcome Dr. Leena Gandhi, who you'll hear a little bit more about in a moment. Can we advance the slide, please? Or am I going to do it? Okay. There we go, next. So this is our seminar series for the upcoming year. We have Luis Diaz, followed by Susan Domchek, David Hong, Jen Margot, Charlie Sawyers, Ursula Matrimonis, Kathy Wu. And next slide. And now I'm going to turn things over to Dr. Chris Chen. And for those of you who don't know Chris, he's the director of our early drug development program at the Stanford Cancer Institute. Chris, want to join us?
Yeah.
Thanks.
Thanks, Steve. So the format of today's seminar will be about a 45 minute talk. We'll save about 10 to 15 minutes for questions. And for those on Zoom, please feel free to type questions in the chat. We'll try to be mindful. And then otherwise, I am very excited to get the chance to introduce Dr. Leena Gandhi. Obviously, immunotherapy has been a revolution in thoracic oncology and other cancers, and Dr. Gandhi has helped lead this revolution. She's ran a number of pivotal studies really advancing our understanding of how immunotherapy can be combined with chemotherapy for the treatment of lung cancer, and how PD-L1 is an important biomarker in utilizing immunotherapy. I think her career is also remarkable for having crossed academia and biotech and large pharma in unique ways. So she got her PhD from Berkeley and then an MD from NYU, and then did her oncology training at Mass General and Dana-Farber fellowship program. And she started her career there as a faculty member, as a thoracic oncologist and early drug development investigator until she moved to NYU to lead the thoracic program. She eventually left NYU to be vice president of immuno-oncology for Eli Lilly, where she led their immunotherapy portfolio. And then eventually going back to academia to head the newly formed Center for Cancer Therapeutic Innovation, which is a unique cross-malignancy novel therapeutics hub, and effectively the phase 1 arm of Dana-Farber, and she was the founding director. And just recently, she left that role to join an exciting company called NextPoint Therapeutics, so she's the chief medical officer based on science being spun out of Dana-Farber. And we're very excited to hear her talk today.
Thank you, Chris, and thanks for the opportunity to speak here. I just very recently left Dana-Farber, actually a few weeks ago, so I will wear my Farber hat to really talk about early phase clinical trials, and I think the new opportunity that they present for accelerating precision oncology in all spheres. So I think, maybe to start, I will just say that I think the paths to drug development have historically largely focused on the what, meaning that lab-based discoveries are really where we consider the breakthrough to happen. We've identified a new target, a new pathway, and the clinical development thereafter is often a series of automatic steps. Maybe the definitive phase 3 trial leading to an approved new therapy is a more pivotal clinical part of that process. But I think that's an evolving structure. Historically, that meant in this context that traditional phase 1s had a very limited role to play, and their only role was really to define safety and to move on to the next step. And even though there have been a lot of innovations in terms of phase 1 designs and how we can better do dose optimization over the past 20, 30 years, I would say that there's still a large dependence on a 50-year-old model for phase 1 development, which is a standard 3+3 design, and where we get to a maximum tolerated dose. This kind of endpoint was developed in the era of chemotherapy, where that's all that really mattered in defining the dose. And therefore, the dosing for both those agents, as well as really everything developed in the last 50 years, has been primarily based on safety parameters. This model also is a model in which, when we really figure out what are the right patient populations to target drugs to and really have the most maximum effect, that happens largely after the course of clinical development. And I think EGFR inhibitors in lung cancer are a very good example of this bedside to bench methodology, where the clinical observations largely that are starting to happen in clinical practice when the drugs are approved, or maybe in the very latest phase of clinical development, identify unique features that lead to going back and looking for particular tumor biology characteristics that might define why those drugs are working well there. So EGFR inhibitors were first approved in all lung cancer patients based on a randomized trial against best supportive care that showed that it was marginally better than best supportive care. But what that meant is that the path to true drug development for EGFR inhibitors was very long, really spanning almost 3 decades. And that meant that there were lots of stops and starts too, from the time of discovery of EGFR inhibitors to even after their approval, since the dose was wrong for gefitinib, leading to its disapproval, then reapproval in a selected patient population, rescinded approval for erlotinib and other patient populations, essentially a very long path to get to the patient population where EGFR inhibitors matter, those who carry mutations in the active site of EGFR. So prospective application of biomarkers to drug development, which really may have started happening with Gleevec in cancer in general, but I think in solid tumors is really characterized by ALK inhibitor development. I like to put this slide up here because it shows the number that we label trials at DFHCC, which is by the year they were started. So this phase 1 trial was started in 2006 and just closed last year, having resulted in at least 4 specific approvals out of that phase 1 trial. But the earliest was really an ALK rearranged non-small cell lung cancer, and this was a patient population that was really defined from these 2 seminal papers after the start of the phase 1 trial. And you can see that the phase 1 trial was not about ALK. It was called a MET HGFR selective tyrosine kinase inhibitor, open to really all comers, planned to retrospectively look at MET expression and try to find a patient population from that initial starting point later on. But once we started to put on ALK rearranged lung cancer patients in 2008, there was a sort of a rapid realization that this is the patient population of benefit. And really the approval for this drug came out of that phase 1 trial. So instead of waiting until phase 3, we were now approving drugs out of phase 1. And I think this sort of was the start of a real cascade of agents in the targeted therapy space, in lung cancer and in many other cancer types, where drug development got faster and faster with the prospective selection of patients into the right trials. So crizotinib shortened that timeline to really 3 years between the starting of putting ALK patients on that trial. I think it's most characterized by the two-year time to approval of osimertinib. And these are accelerated approvals that are coming out of phase 1, but approvals resulting in clinical use nonetheless. So enrichment of the appropriate patient populations in phase 1 really provided an opportunity to accelerate drug development. You can really differentiate, by trying to select or enrich patient populations, who are the responders versus non-responders and why, even in the context of that clinical trial, and get to more rapid proof of concept, ultimately leading to accelerated approvals, which I think is really the new model for oncology drug development today, that ushered in this new era where we no longer actually have 3 separate phases of clinical development, but we really much more often put phase 1 and 2 trials together into a proof of concept initial trial that is the pivotal trial, leading hopefully to accelerated approvals, use of therapy, and the phase 3 trials are now the more of an afterthought to hopefully confirm what has been observed in those earlier trials. I think the advent of immunotherapy, at least the new advent of immunotherapy, because certainly immunotherapy has been in development probably for more than 100 years in oncology, but the new advent of immunotherapy, which I characterize as the PD-1 era, kind of highlighted that things aren't going to be as simple as they are in the targeted therapy population, where there's not a fixed genetic change and a clear-cut way to selectively enrich patient populations. This incredibly simplified diagram really highlights, I think, the fact that there are multiple inputs to a tumor immune response. It's an integration of many different signals, many different cell types, so lots of determinants of that immune response, not just one primary driver of oncogenesis. And even though PD-L1, I think we thought of as a obvious biomarker to look at, it was by no means thought that this would ever be a really very good biomarker. It was imperfect in many ways. One, it's one ligand. It wasn't even the only ligand for PD-1 receptors, but there were also PD-1 receptors in many different contexts and PD-L1 expression on many different cell types. So there was an initial concern that we don't know where to look for PD-L1. Is it on tumor cells? Is it on immune cells? And the assays that were being used in clinical development were all different, that we worried actually how different they were and that they would never have concordance. And because there is such geographic expression of PD-L1 in tumors, that the selection based on PD-L1 would never be accurate because of sampling error. And in fact, in the phase 1 trial, what we really saw is that the differentiation for response, even if you just try to look at the most simplest aspect of PD-L1, which is tumor proportion score, a proportion of tumor cells that are positive, was really very low. It's at 37% in this initial cohort versus 10% in those who were completely negative and 37% in the highest group. But what was really noted is that in a validation cohort, you could clearly differentiate longer-term outcomes, and that's what really made PD-L1 a good biomarker. Not its validation as a response biomarker, but as a long-term outcome biomarker. And that's what really ultimately led to accelerated approval of pembrolizumab on the basis of a phase 1 study, something we didn't think could really happen in the context of immunotherapy. Well, I would say maybe things that other people thought couldn't happen in a phase 1 study. From a lung cancer point of view, this is where people came on board with immunotherapy, because targeted therapy we can understand. And if there's a way to target therapy in immunotherapy, then we're all gonna be on board. But what was striking about this development was the integration of efforts in a phase 1 study. So this was an all-comers phase 1 study. Ultimately, the size of the KEYNOTE-001 study was 2,000 patients, phase 1 study. And this accelerated approval in lung cancer came really through a validation of response. So accelerated approval is mostly based on response in really a group of only 61 patients. Mind you, out of a group of 500 patients, lung cancer patients on that phase 1 trial, so a large phase 1 overall. What we've learned since then is actually how hard of a biomarker PD-L1 truly is, that it has not only different places of expression, but different predictive value based not only on the level of expression, but based on the location of expression, that the predictive value is different in different tumor types. We know in breast cancer, PD-L1 is not commonly expressed on tumor cells. There's not tumor intrinsic expression, but likely it's primarily an adaptive immune response, driving whatever small patient population gets benefit. And that's why immune cell expression is really the biomarker of interest in breast cancer, in melanoma, and in renal cell carcinoma, where there are very high levels of PD-L1. The predictive value is very, very poor, and it's really not used as a clinical biomarker at all. Even in the context of lung cancer, we know that there are not histology-specific context to this differential use of a biomarker, that there are some lung cancer patients who do not have tumor cell expression but may have immune cell expression, and often these two populations are often separate. This is actually from a phase 3 trial of atezolizumab in lung cancer, showing that populations where there's high levels of tumor cell expression are often mutually exclusive with those populations that high levels of immune cell expression. That's depicted here versus the tumor cell expression. And that these are probably two different phenotypes, one of an adaptive immune response that can be really revved up in the presence of PD-1 inhibition, and one where you're overcoming tumor intrinsic immune suppression by tumor cell expression. So two different biologies, same histology, two different uses of the biomarker, and not a clear way to how you might integrate those. Still today, actually, in lung cancer at least, tumor cell expression is the dominant way of using this biomarker, because we actually have such variability in the immune infiltrate in lung cancer biopsies that it's hard to actually rely on immune cell expression as a reliable clinical biomarker. But for a subset of patients, that's probably the only relevant biomarker. What we have learned through all the data, not only from PD-L1, but from other potential biomarkers like CD8, like TMB, like gene expression signatures that are reflective of T effector cells, is that an integration of biomarkers has more predictive value than any one biomarker. What we haven't really quite learned is how to integrate those different biomarkers in clinical practice, but there are more and more trials that are using an integrated approach in clinical studies to select patients to actually define what patient populations are going to get the best benefit, particularly for combination therapies. Ultimately, I think Keynote-001 was not the model for phase 1 studies going forward, because what happened really as a result of Keynote-001 is that the FDA really cracked down on doing those 2,000 patient phase 1 trials. In 2018, they limited expansion cohorts to really a size of no more than 40 patients, which means that it is really a challenge to both indication exploration as well as biomarker exploration at an era where not only are we doing more complex biologics in terms of multi-targeted therapeutics, but there's much more combinations happening in immunotherapy drug development. So it's a much more complicated questions that you have to ask in phase 1, and now we have to do a lot more with a lot fewer patients. That's really, though, I think, a challenge that as a field is where there's an opportunity for both academic institutions to really partner more effectively with industry in how do we do these kinds of studies, how do we actually make the most and get the most out of the fewer patients that we're able to test in phase 1 studies to really know the right path forward. I think this is an opportunity to use maybe what the, quote, newer phase 1 design concepts have been percolating for the last 20 years. A lot of these newer approaches, such as defining an optimum biologic dose, are not new at all. They've been around for a very long time, but they're not used very effectively, particularly in the targeted therapy model, where there's a much straightforward way to select patients, do the trial, get the results, get approval. But optimum biologic dose is really, again, was primarily based on safety considerations that in the era of non-chemotherapy agents, it's more important to define where are we actually getting the right biology happening, where are we getting target inhibition, as long as there is relative safety. Overall, although this has been promoted for many years, in terms of actually changing phase 1 design, this has had a very limited impact to date, primarily because the definition of optimum biologic dose varies from trial to trial, from target to target, and in vitro estimations of what an IC50 is really not the same as in vivo. It might not be the same in animal models as it is in humans, not the same in tumor as it is in blood. So it's still not a common primary objective, at least until now, and it rarely has formed the dose decision basis in most trials to date. I think that might be changing, and I'll come to that in a moment, because of a further shaping of the regulatory environment. But the other idea that came out of safety, which is again not a new idea, but is starting to influence more of phase 1 dose design, is the notion that traditional models of toxicity cannot really define what our starting dose level should be. So the NOAEL, or the no observed adverse effect level, was always based on animal toxicology models, where let's start at 1/6 the dose of where there's really no observed adverse effect in mice or in non-human primates. But that means you have to have a relevant animal model. What we have learned in the era of immunotherapy is that there are frequently no relevant animal models to not only model toxicity, but efficacy as well. So that really led about, and again, this is not new, this is really from 2013, where MABEL approaches have been promoted by regulatory agencies, but that's really trying to define the minimum active biologic effect level as a starting point. And again, this was not really trying to get at the biology of the tumor when these regulations were made. It was really about safety. And it primarily came about as a result of a toxic death that happened with a CD228 superagonist. But the idea is that if we can't model toxicity in an animal, we likely need to be starting a lot lower. So let's take the comprehensive data that are generated in preclinical models, and that might be in vitro for a lot of immunotherapies. Where does T-cell activation occur? Where does T-cell proliferation happen? At what level? Just in an in vitro system, what surrogate models can you use for efficacy, and what dose responses can you generate to see where that starting dose may be. And again, the idea behind this is that typically the MABEL starting dose is significantly lower than the NOAEL. So this was really a preservative measure for safety, that we would protect more patients by starting at lower levels, just like optimum biologic dose might protect more patients. But ultimately, really what the results of those goals are is that if we're really focusing on where the biologic effect happens, that means we're focusing more on the biology. That means that we're really actually trying to say that what's needed to get into the clinic is actually pharmacology studies and actual pharmacodynamic studies that tell us when we're actually doing what we think we're doing with the drug, and using that as a starting point, and also the point at which we might, how we define pharmacodynamics in the clinical context. How do we define what we're doing with the drug in patients, when if we can do it in models and we'll use those same kind of assays in patients? It really has led to a more mechanistic-based design. And I would say MABEL approaches are now sort of the common and defining mechanism to develop immunotherapeutics in the clinic, regardless of safety parameters. We often think of immunotherapies actually as generally safer, CD28 superagonists notwithstanding, than many other types of therapies, and that the safety risks aren't as high for patients. But what MABEL dosing has allowed us to do is be more actually scientific about how we do phase 1 trials. And to me, actually, that's what's really the appeal of immunotherapy in phase 1. I think you heard that I did a PhD before I ever went to medical school, and ultimately knew I didn't want to be doing lab-based research. But what I really like about immunotherapy is you have to do the experiment in the patient. You have to actually understand the mechanism in a patient-centered experiment that you can't really do in other models very frequently, particularly for newer immunotherapy agents that target cells that we can't even model in mice, the myeloid component and more components of innate immunity. But the more recent development of Project Optimus really has actually, as of this year, January 2023, resulted in draft guidance that basically says no more MTD designs, that we're not going to base decisions about drugs and how drugs work and how drugs can help patients only on the basis of safety. Now all drugs, targeted therapies, chemotherapies, antibody drug conjugates, and immunotherapies really have to use biologic bases for how we might develop these drugs in patients. And that means a totality of data around safety, yes, around dose exposure, but also anti-tumor activity and pharmacodynamic evidence of anti-tumor activity should be used to select doses. It has also mandated that we do more dose exploration and comparison of different doses very early in development. So although in 2018 we were told that we could no longer do large phase 1 trials, we've almost come full circle, because now what we're being told by regulatory agencies is you really need to figure out what's happening in phase 1 before you can move on. So I think it's actually given us license again to do the kind of robust biomarker exploration and understanding of biology in phase 1 before we move any further. I actually had a conversation recently with Hal Barron, who used to lead R&D at GSK, and he told me that he was asked by his investors, how can we accelerate drug development and how can we accelerate drug development? And his answer was, we need to slow down, because really what we need to do is actually learn and understand how the drug is working before we start using it in a widespread way in large patient populations. So really that regulatory environment has helped shape, I think, our ability to do more in phase 1 and with more patients, because now we're really going to have to apply all these sort of models that were cast aside over the last 20 years and really do real-time dosing decisions based on biology. That means adaptive modeling, continuous reassessment, randomization in the context of phase 1 trials, and really trying to understand relevant target inhibition, and really, which means that we're actually going to be able to get more data because we have to do more tumor biopsies in the context of these trials to actually understand the biology of how to move forward. So ultimately, I think what this has resulted in is that now phase 1 trials have gone from the lowest on the totem pole in terms of role in drug development to really like this is maybe where we're going to make the breakthroughs, because now we have to understand proof of mechanism, especially for immunotherapies, in the context of a patient experiment, and we're going to actually have to do that in the context of a phase 1 trial. So we don't need just the what, but we also need the how, and the how really happens in phase 1. So to me, that's exciting, because having been in industry and academia, I think this really opens up where there's a lot more opportunities for partnership. There's an increased need for translational data in the human context. There's an increased need for more complex translational work, which means we can't rely on sort of standard biomarkers. Maybe we're looking at completely novel biomarkers. Maybe we really need to have complex integration of biomarkers, and that means we need academic partnerships with people doing cutting-edge technologies to look at multiple order inputs to a tumor immune response in the same way. And that means that we need all those things in the context of a phase 1 because they actually have to guide dose escalation. That becomes particularly important when you think about combination therapies. And I think this has been a holy grail for a very long time. This is a slide from Tony Ribas from 2012, talking about the potential for immune therapies in combination. What can we do beyond the often 20% threshold that we see in terms of response with PD-1 inhibitors? We're still struggling to find that. I think chemotherapy was an easy place to start, although probably the most complicated place to start because chemotherapy has very complex and pleiotropic effects on tumor immune responses, some negative and some positive. Ultimately, the trial that was done, because it was again in the era of speeding up, it was 2018 actually, that we were doing a lot of these, was that we knew that pembrolizumab combinations or anti-PD-1 would both enhance the anti-tumor effect of chemotherapy in an immune response setting and also impair the promotion of reduce the negative effects of chemotherapy and an anti-tumor immune response. But ultimately, the trials that were done really didn't take into account what we already knew from preclinical data, which is that chemotherapies are not all the same, and chemotherapies actually have different impacts on the tumor immune response in combination versus single therapy. Pemetrexed, which was the backbone of the combination chemotherapy used in lung cancer with immunotherapy, certainly in preclinical models, this is in syngeneic mouse models looking at volcano plots of immune cell expression of T-cell activation markers. And you can see there's a much more robust induction with pemetrexed alone than with pemetrexed plus carboplatin or compared to other drugs. Ultimately, though, the trials were really informed by what was used in clinical practice. It was not clear that we were going to see what we saw, which was there was a really big difference. But what we don't still know about this response is who are these patients that are getting this benefit? Is this just an additive effect? Because the only thing we learned from these large phase 3 trials was that PD-L1 was still predictive of who was getting benefit. Does that mean it's really just an additive effect and there are not synergies that we're really capturing in this group? I think these are still somewhat unanswered questions. I mean, what we've seen recently in the field is a lot of immunotherapy, chemotherapy combinations, but fortunately, I think that this is a transient stepping stone to where we're really going to extend the benefit of immunotherapy, which is in immunotherapy combinations. How do we really boost an effect beyond PD-1 by other kinds of stimulants to the immune response or other kinds of de-repression of the inhibition of immune response? So today, novel IO combinations really dominate the clinical trial space in oncology. And there's also, if they're not combinations, then they're multi-targeted therapeutics combinations in one molecule. So these are where we're going to get the opportunity, I think, in the new regulatory era to really apply what we've now learned, which is that we can't wait to figure out the right biomarker strategy, but we have to be thinking about that up front. And that means we need really robust and really comprehensive biomarker work in phase 1 trials to really understand the biology before we move further. But fortunately, I think there's a lot more effort in that space, including here at Stanford, because I know you're a part of the CIMAC network. But I think that kind of in-depth immunoprofiling from phase 1 samples, where we're really maximizing the use of biopsies that we get from patients who are getting multiple biopsies through the course of the trial. If we're not getting information from those biopsies, we're doing no favors to those patients either. So that kind of intensive work is, I think, really critical to how we advance the next step. Unfortunately, I think CIMAC is one example, but there's really multiple academic industry partnerships that are working towards this goal, and I think hopefully we're going to start to see that really translate into making a big difference in what agents move forward and what agents don't move forward. There's certainly been, it's certainly right now, because we're still learning in that space, I think a lot of agents that move forward and fail spectacularly in phase 3, and then some that win in phase 3, like relatlimab and melanoma, that almost seem like luck because we didn't really learn what they were going to do in phase 3 from the phase 1. But those studies, which cost $500 million, maybe we could spend some of that $500 million actually in biomarker analysis in phase 1 and ultimately both save cost and move faster to get to the right place. The other critical piece that I think will be important is actually redefining who are phase 1 patient populations and how we target drugs. So I think in the era of targeted therapy, there was a real pendulum swing towards histology-specific trials because ALK rearrangements are really largely only found in non-small cell lung cancer, lung adenocarcinoma, same for EGFR mutations. With the advent of immunotherapy, since we all have an immune system and all tumors interact and interface with the immune system in some ways, I think we're seeing a little bit of a pendulum swing back to where the era of chemotherapy was, which is that these agents can have effects in many different tumor types. And actually now our challenge, because we have the ability to get data, which we didn't maybe in the original chemotherapy era, is to figure out who are the right patients to actually get that benefit. This is a study actually from UCSF, but I think it highlights what a lot of us are trying to do in the metastatic space. This was a study actually taking resected tumors, sorting cells, so sorting TILs out of those tumors and then doing bulk RNA sequencing to define multiple immune-specific phenotypes. I would say in the initial era of immunotherapy, we could define maybe 2 phenotypes, those that were PD-L1 positive, those were not, those that were tumor mutational burden rich, and those that were not, and those that were CD8 infiltrated. And that was sort of the extent of it. Now we're getting to really the integration of big data to really define signature-based phenotypes that I hope will form the basis for who goes on to a particular clinical trial. That it's not lung adenocarcinoma that doesn't have an EGFR mutation, but it's really a patient that has a myeloid-centric DC2 bias that's going to be a very appropriate patient for a myeloid targeting checkpoint inhibitor. And where we'll really get to how do we put the right patients on phase 1 trials so that when we do this intensive biomarker work, we're actually going to be able to differentiate responders from non-responders rather than looking for a needle in a haystack, which is what often happens when you put an unselected patient population on phase 1 trials. So the other piece that I think that is a challenge for us going forward is that the drug development landscape has really exploded in oncology. This was from just a recent search on clinicaltrials.gov. There are over 18,000 active trials for cancer right now. Almost a third of those are phase 1 trials, and really almost 1,500 new phase 1 trials each year, which means that we're, as institutions across the US, competing for smaller and smaller pools of patients in an era where, fortunately, there are more and more successful therapies that have extended standard of care options and slowed down referrals to phase 1 clinics. So I think it presents unique challenges. And to me, this is where phase 1 is sort of poised to change and pivot about how phase 1 works both in academia and in industry and designing trials. So those are sort of the setup for how I approached coming into the Farber and building a phase 1 program. And maybe the last piece is that one of the advantages, maybe less a challenge, that the biomarkers that we're now routinely using in clinical decision making are now getting more and more licensed to be used for clinical trials too. So, although genomic testing, we can argue, has not still had the penetrance that we would like it to have, it is still getting more and more each year and has actually gone from 20% to 60% in the last in a 5-year time frame in non-small cell lung cancer. I think that's actually increasing more and more rapidly with recognition that there needs to be more prevalence of doing that kind of testing. But the other is that there's now sort of ASCO guidance that really emphasizes getting away from histology in terms of looking at genomic biomarkers, that the more we look, the more we find that actually you can find EGFR mutations in other cancers other than non-small cell lung cancer. You can even find these altered immunophenotypes in different cancer types that are not histology specific. And it provides a framework for testing most solid tumor patients on the basis of tumor agnostic therapeutic approvals, which we're seeing more and more of, for NTRK, for example, for RET, We're seeing more and more targeted therapy approvals come in a non-histology specific way, and I think that's opened the door for more non-histology specific testing. But it also, this is from an ASCO provisional clinical opinion last year, but it also has opened the door for saying, if we're going to be testing all these patients, let's use that information for how we actually find patients for the right clinical trials. So these were the factors sort of, to me, that led me to come back to the Farber to actually help set up our phase 1 program there, which we call the Center for Cancer Therapeutic Innovation, but the goal of which is really to accelerate the development of novel therapeutics, really by doing more tailoring to specific patient populations from the very beginning. And to me, that means what the opportunity for phase 1 is to serve as that hub in a central translational midpoint of drug development where there's historically been gaps, because either the initial discoveries are made in academia, the R&D is done in industry, the clinical trials come back together in collaboration only in the later phases. What we know, or at least what happened at the Farber, and I think this happens probably everywhere, is that there's often very robust clinical collaborations that happen with experts in the field, that people will come to Dr. Wakely to do really pivotal phase 3 trials in lung cancer, and there might be really great collaborations happening in the setting of discovery science in people who are experts in particular disease biologies. In telomerase, for example, there's probably robust discovery collaborations happening with companies in academia. There even could be some target validation collaborations happening, but where there's often a big fall off or drop off is in the place where clinicians and scientists come together. And in most institutions, there are clinicians and scientists working together all the time. And I think it really is a place where, at least at the Farber, we really wanted to serve as how do we bring people together into this hub so that we continue this wheel, the bedside to bench observations and the bench to bedside, so that we're actually making this connection each piece of the way through. And that was sort of the goal in setting up a more united effort around phase 1. Prior to that, at the Farber at least, and at many institutions actually, there had been separate efforts in immunotherapy versus non-immunotherapy. Those fields are coming together, especially with targeted tumor antigen targeting therapies that bring T cells into a tumor. So there's many different ways that those fields are coming together, and it didn't make sense from a patient perspective to do that any longer. And one of the other things that to me was very important about doing that is, one of the critical pieces I learned in industry is that phase 1 has dependence on speed in a way that no other phase of clinical development has. Biotechs live and die actually on the basis how fast they can actually conduct their phase 1 trial. And I think that's part of why phase 1s have often been short-shrifted and only been as minimal patients as possible, just get enough safety data to move to the next phase. Is because we don't have time. We just have to move to the next phase. Now that we say that phase 1 has a critical importance, it means that we need to spend more time there, but getting that started is of critical importance. And the operational components of doing that on the academic side often are a disconnect with industry. But I think there really are opportunities, and that's one of the things we tried to focus on, to accelerate operationally how we can get into phase 1 and get phase 1s open, even despite all the increasingly demands of a regulatory environment. But our other goal was to become a translational hub for cross-malignancy research. At least at the Farber, our portfolio was about 100 active phase 1 trials, which meant that there was a lot of opportunity to get tissue to actually define some of those new phenotypes that are histology independent for the next phase 1 trial. So about 75% of our portfolio required biopsies, which means there was really an opportunity to get tissue to inform drug development in the right patient populations, rather than what's been used by the community at large for the last 20 years, TCGA, which is largely resected specimens from early stage disease, which doesn't necessarily reflect the tumor microenvironment of the patients you're actually using these treatments in. So for us, the goals were really around developing a centralized and specialized team, meaning that our oncologists were really all disease specialists because they brought to bear their clinical expertise. What are the right biomarkers? How do we use biomarkers in the clinical context? And how can we apply them to clinical trials? And how do we take things forward faster into phase 2 and phase 3 settings and specific disease indications? Operational efficiencies and really the translational research. This is our group at the Farber, who represent at least all of solid tumors and translational investigation. And one of the first things we did was to really establish a cross-malignancy clinical pathologic database to really actually make use of the biopsies that we were getting for these therapeutic trials to actually generate data that will inform the next trials. And that meant not banking tissue. This was not a specimen banking protocol because, as you all know, if you work in the clinic, we get really small biopsies from patients undergoing phase 1 clinical trials. They're often patients who are getting really small needle biopsies, and a company running the trial will take half of that. Sometimes we never see the results of that work that gets done in the company, but there's really an opportunity to get tissue to do other work, but that means parsimonious use of that tissue. So what we've instituted is sort of immediately from the biopsy, when we send tissue to a company or an industry sponsor, we're getting additional tissue that we process immediately for DNA, RNA, and for the most common malignancies in phase 1, colorectal cancer, lung adenocarcinoma, and ovarian cancer, we're processing for single-cell RNA sequencing, so we can actually get a pooled database of really more in-depth profiling across these patients, as well as spatial immunoprofiling. We're also getting blood that we get methylome data, ctDNA data, and HRD data from. And our goal in this is not to really link this data with outcomes on those specific clinical trials, but to really define the context to develop new agents in, define the new myeloid phenotype for the next myeloid inhibitor, to define the new phenotype for the next DNA repair combination, and so on and so forth. The other reason that we're sort of getting this tissue, but also gathering clinical information, is to actually use biomarkers to inform clinical trial selection. And this has been an effort of integration going on at the Farber for some time. I think it's happening at a lot of institutions. But I think the goal in doing this is to say what we're doing actually in lung cancer right now. I was talking earlier with somebody about the fact that, we have a lot of tools or information that we get in the clinical setting that we use on a patient-by-patient basis to make decisions. We can also apply those to actually clinical trial selection, particularly if you have large clinical trial portfolios for hypothesis testing. So at the Farber, we've been doing next-generation sequencing for about 12 years now, so that means we've done at least 50,000 tests. Some of those have a little bit wider panels versus not, but we have a lot of data actually that can be used to inform thoughts about clinical trial selection. And this has really broadened from initially used primarily in lung cancer to clinically billable tests across many different malignancies. So we're doing this on a ever-increasing basis. So initially it was a philanthropy-supported effort. Now it's actually clinically billed in many different indications. So about 6 years ago, we started a computational platform to use genomic information to match patients to clinical trials. And I will say this has had a checkered history because a lot of the data sometimes is not in the right context. Sometimes it's from their diagnostic biopsy, not when you really need that information to put a patient on a clinical trial, or that patient, because they're referred to a tertiary cancer center, may never come back and you may not really know when the right time to use that information is. So MatchMiner 2.0 is an initial effort that uses, on top of the genomic data, an AI algorithm developed by one of my thoracic colleagues, Dr. Ken Keel, to identify trial-ready patients on the basis of machine learning for radiographic reports, where we use words like progression, increasing, and other things to determine when a patient is actually progressing on therapy and might be, quote, trial-ready, to moving on to a trial. And this has really enabled us to narrow down the list of patients where we actually do more intensive trial matching using actual humans in a way that we can actually select patients onto clinical trials. In addition to the genomic information, about 4 years ago, we started doing immunoprofiling for patients in a small, I would say 1.0 beta version way of doing multiplexed immunofluorescence, at least for some common T-cell markers, and doing this across solid tumors and validating the same assay to be used across solid tumors. So this was an effort really started by Scott Rodig in the pathology group and in the Center for Immuno-Oncology. And now we are routinely performing this immunoprofile test, which is 5 biomarkers on patients across indications who have an indication for anti-PD-1 or PD-L1 therapy. Our goal is, and our hope is that, just like for NGS when we started it, we were able to anchor the cost of that to be billed to insurance by EGFR testing. Here we hope that we might be able to anchor that billable test with PD-L1 testing, which is used in many of these different contexts. we've now been able to integrate that genomic information, that immunoprofile information with genomic information using the match minor algorithm. So we're now trying to say, look, like we have this data source, how can we use this information to actually select patients for different clinical trials? So we're doing this also by whatever other data that we think will be useful that is gathered in piecemeal ways. We do HLA typing on a lot of patients for selected clinical trials that can be used for a lot of future clinical trials as well. So that's part of our database gathering, as well as tumor antigen expression, although HER2 is not used for clinical decisions in many different malignancies outside of breast. It's actually overexpressed in many different contexts, and as you all know, there are many different tumor antigen targeting agents, antibody drug conjugates, cytokine fusions, and others that will use a common tumor antigen like HER2 to actually select and maybe implement a drug for clinical trial testing across different tumor types. We also get increasing data around RNA expression, both from the use of commercial platforms like Tempus and other efforts within different trials across the Farber. So we're trying to integrate all of that information in a way to sort of prospectively do hypothesis testing of how can we integrate the use of these biomarkers in clinical trial selection. Our goal is to use that outcomes data relative to historical outcomes for phase 1 trials as a first pass to see how much that integration can really be used in a randomized testing fashion in the future. But just to give you an example, these are patients that I took care of in the context of phase 1, and just to take two similar patients, these were both women with very limited smoking history with metastatic lung adenocarcinoma with KRAS mutations. The first is this patient, a 73-year-old woman who had had progression on carboplatin, pemetrexed, and pembrolizumab with stable disease as her best response. It lasted about 6 months, which is the PFS, or the progression-free survival, typical for chemotherapy. So another patient where we really don't know the impact of immunotherapy in this context. She was referred for KRAS-targeted therapy, but because she had immunoprofile data, which also showed very high levels of PD-L1 expression, although she didn't respond to PD-1, but significant CD8 infiltration as well. To me, this was the patient that, although she had a KRAS mutation, she likely had a very immune-primed microenvironment for combination immunotherapy. So I put her on a combination of an anti-PD-1 inhibitor with a novel anti-angiogenic. She had a very major response, even though she was progressing so rapidly, actually, that her disease burden progressed during the screening period. So we had to rescreen her on the day of starting therapy because she was progressing so quickly. But she had a major response that lasted over 6 months. Eventually did go on to a KRAS-targeted trial at a later date. But for a different patient who had a very similar profile, she had a different KRAS mutation, but also a non-smoker with metastatic lung adenocarcinoma. She was also treated with carboplatin, pemetrexed, pembrolizumab with stable disease. She came with an immunoprofile that really had 30% CD8 infiltration, so less than most of the comparable tumors in that data set, and also a significantly higher FOXP3 expression or Treg expression, so regulatory T cells that might impair a tumor immune response, for which we didn't have open trials. And for her, I put her on a more, quote, KRAS-targeted trial, targeting the KRAS pathway with a novel ERK inhibitor. Which she also had same kind of picture, a significant response to, lasting more than 6 months because she progressed in a time frame typical for a progression on ERK inhibitors, where we know BCL2 expression is overdone. She went on to actually to a MEK BCL2 combination, which she also responded to. But just some examples of how we're trying to actually prospectively enroll patients using clinical biomarker data that we hope will be able to validate for actual prospective testing in the future. So the goal, at least for us in reorganizing the structure of phase 1, was to really maximize those phase 1 opportunities for translational collaboration, for clinical biomarker use, and for biomarker testing to get to the next phase of new trial targets. And I think that's really the goal and the opportunity for phase 1 everywhere, which is that we know a lot about how to do the what. Now, if we can actually improve the how, that's where we get to the faster breakthroughs. I think the opportunity is really for clinical trial collaboration, particularly with industry academic collaboration, is very unique in the phase 1 setting because it's often much fewer institutions involved. They're often really more partnered experiments where you can do translational work together. That's often really hard to do in the context of a phase 3 trial with 70 different international sites. In a phase 1 trial, there are often 5 sites, one of which really is good at single-cell RNA sequencing, one that might be really good at flow cytometry, where you really have opportunities to do a real partnered experiment. And get to proof of mechanism, which is really how you get to biomarker validation and how you get to accelerated approval. So I will stop there. I just wanted to thank the group at the Farber that I have worked with for a while, and hopefully they're going to continue this work going forward. Thank you.
Yeah, I'm fascinated by our use of the immunoprofiling. Can you speak a little bit more about how you introduced it clinically, how you operationalize, how you pay for it, and how are all patients screened for this?
Yeah, well, I will say it is a little bit of, it's undergoing ongoing evolution because our pathology department recently merged with Mass General's. But up until that merger, I would say what our goal was to model what we did with NGS testing in the setting of this immunoprofiling. So what we did with NGS testing is it was initially a fully philanthropy-funded effort. We used it sparingly, actually, where we thought it would have the most impact, and actually that was in the area of non-small cell lung cancer, because we knew, even though ALK inhibitors weren't approved, that we wanted to find ALK-rearranged patients. We wanted to find BRAF mutated patients because we had ongoing trials. We wanted to find T790M EGFR mutated patients for trials that we were running. And as all those things became validated, it was very easy to say the cost of that NGS testing, which has come down a lot, is really no different than the cost of doing EGFR plus BRAF plus ALK. So we were able to get clinical billing and insurance to pay for the cost of those tests, which then actually enabled philanthropic efforts across other tumor types. And as more and more tumor types have gotten those things approved, that again sort of moves to a billable insurance paid for model. So because DNA repair inhibitors are now actually approved in context of HR deficiency, and you need NGS testing to do that. It's really made NGS testing very widespread across different tumor types, at least at Dana-Farber, and that's the goal of the immunoprofiling as well. Many tumor types actually have PD-L1 inhibitors approved, or PD-1 inhibitors approved in those settings. We hope we're getting more information beyond PD-1, but we hope that as we validate the assay and we use it more, that we will be able to transition it to a billable test in the setting of lung cancer, for example, where PD-L1 testing is standard. And then, again, initially with lung cancer, and then probably using the fact that we're testing both PD-L1 and PD-1 and on immune cells and cancer cells, that we'll be able to sort of bill for it across different indications where CPS or PD-L1 testing is used. And our goal actually with immunoprofile is to continue to expand that, because that is a moving target. It's only 5 biomarkers. It's very T-cell focused. From a trial perspective, we would like to have a lot more information than that, particularly around the myeloid cell component. Right now, the CD8 component, I would say, is the most useful. And up until our pathology departments merged, MGH separately used to do only CD8 testing. So they'd integrate CD8 and PD-L1 testing, but similar idea. As we reorganize the pathology departments, our hope is that we can expand the immunoprofiling testing to be across higher levels of tumor types. But that's also an expanding role. So right now, the goal is to do it in patients where PD-1 or PD-L1 inhibitors are approved. But as you know, that is an ever-expanding group of patients. So it didn't used to include mesothelioma, and now it includes mesothelioma, and so on and so forth. That's going to, I think, increasingly encompass more and more solid tumors.
I have a question about your trial you mentioned in the beginning, 2006.
Mm-hmm.
I didn't quite understand. You said that the trial was open for a long time. There was MET and some other inhibitors, but then you pushed the ALK inhibitors through that. How does that work? I mean, how do you create like an umbrella protocol of some kind? You can put multiple.
Well, I think I use it as an illustration of it illustrates, I think, the constant iteration that phase 1 trials have. So that's an example of a good partnered experiment. So the company doing the trial was Pfizer. Had initially billed it as a MET inhibitor. It was the initial inclusion-exclusion criteria were all comers. As ALK patients were defined and we could actually test for them, we started putting them on trial. And as we started to see responses, Pfizer amended the trial to really have dedicated cohorts that were only for pre-selected ALK-positive patients, which is how we got an accelerated approval in ALK-positive patients. Crizotinib is now approved from the same phase 1 trial in MET-amplified patients. And this is across different tumor types, got approvals from the same phase 1 trial, and also it's been demonstrated to be validated for exon 14 skip mutations in lung cancer, although there are other drugs that are preferable in that setting, but it was used sort of as a branching point for dedicated patient-selected cohorts. Since we constantly amend phase 1 trials, and it's a constant evolution of the trial as we get more information, it's an easy place to do those kinds of iterations, and that's why it's lasted so long.
Hi, yeah, thanks. So following up on Chris's question, in addition to kind of the cost of how you do this additional profiling, have there been barriers to negotiate with companies about doing those extra biopsies for patients on clinical trials and then doing extra work? And then the other half is you've seen the industry side of things. Oftentimes our perspective is they collect a whole lot of samples and we never see any correlative data from what's going on there. And so as siloed as we are in academic centers and getting together with a couple other people to collaborate and publish a small data set, industry is even more so, it seems like, of holding on to the libraries of samples.
Yes, and I'm so sorry. I think
solve that?
I think I'm supposed to be repeating the question, so I apologize for not doing that, but I'll just sort of restate that question. So how do you actually collaborate with industry around getting additional tissue to do other more kinds of non-trial specific questions, and how do we actually get data out of all those specimens that are collected by industry? I think those are two separate issues. So the first one is really, you don't need any collaboration with industry, right, to get extra tissue. That's something you're doing as a completely separate protocol within your own institution. They never need to know. That's really none of their, it's nothing to do with them. Just like you probably have protocols, or what we had as protocols that allow for research from clinically acquired biopsies. That's the umbrella protocol that we use to collect additional tissue. That's not funded by industry. That analysis is not funded by industry. Those are funded through other grant mechanisms and other internal funds to do that kind of work and build up that database. What we hope to do, because this is an effort that's just getting started, is as we get more information about, let's say we get a data set of single-cell RNA profile from 50 tumors across 3 different tumor types, and we characterize a certain phenotype that we think would be very appropriate for a new target, then we have that data actually to go and collaborate with a particular company in that space and say, this is where we think you should be doing this trial, and this is how we think we have data to support prospective selection of patients for this trial and actual design of the trial on the basis of this data. That's, I think, different than questions that are trial specific, which in theory is what industry is using their portion of that tumor biopsy to interrogate. And where does that data come from? So I think there's 2 ways to lose that data. One is from big pharma trials. So you're right, I think that big pharma actually have more silos even than in academia, because different study teams will be studying one particular combination or target in one particular disease, and they never want to share that data with anybody else at the company who wants to do a new combination elsewhere. So there's often internal company struggles about how to share those samples, and if they're not pre-specified in the consent form, you actually can't. That's something I think that Merck has done very well, because they have this kind of blanket consent for future biomedical research to ensure that they can always do iterative research on any samples that they collect. They do hoard data, though, Merck, as you know, like many companies. Sometimes it's because they haven't done the analysis. Because honestly, once they get to a certain endpoint, they pivot very quickly, don't bother with doing the more intensive analysis that some of us might want to do outside of that. I think there are a lot of scenarios, unfortunately, especially in small biotechs, where they don't get that data because they can't ultimately, because they're kept, the heat and all the timeline issues are kept on by investors who only care about clinical responses. And if you get one response, then you must move to a phase 3, rather than actually try to understand, well, who's responding to TIGIT versus, PD-L1? We actually still don't know that, even though there probably are patients. But instead of you doing that intensive work, they have to use whatever resources they have and whatever money they have to do the next trial. So they start to pivot and don't ever do that analysis with samples. I think what could be done a lot better is actually letting academics make use of those samples. And I think there is more of that work happening now because people start to do phase 3 trials and see that they aren't actually getting results where they had expected because they got like 2 responses in a phase 1 trial. And that doesn't necessarily translate into large patient populations getting benefit. And they do have to go back and do more work. So I think there are more efforts in that direction. And I think that's where these academic industry partnerships can really make more impact from the beginning. If you plan these things prospectively, you have much more of a chance of actually getting them done. So the CIMECs are actually, I think, a great way to do this kind of work in immunotherapy combination trials and novel immunotherapies, but it's often restricted to NCI-funded trials. I think actually there's an FNIH PACT consortium, which is even better because they do it across more industry trials, but they are only partner with large pharma. I think there's still opportunities to do this in a much better way with small biotech, and that's where I think institution biotech partnerships are really ripe for the picking, particularly in environments like Boston and the Bay Area, where there are a lot of small biotechs right in your backyard, and there's a real opportunity to do that and really plan it prospectively. That's, I think, what a lot of biotechs are trying to do in the Boston area, and I hope we're going to start to see the results of that, because if you plan it prospectively, then the tissue samples, as soon as they're collected, get into the right hands of academics actually who care about making this happen. And I think the reason for industry to do that more is that what industry is constrained by is regulatory obligations. So if you don't have enough of a validated biomarker that can be used in data that will support ultimate approval, you can't use it, right? So there's all kinds of regulations about what you can actually use prospectively in a trial. Just like for CTEP trials, you have to have the integrated biomarker, which has to be more validated than exploratory biomarker. I think there's a lot more room to do that in IITs, in academic partner trials, when a lot of the biomarker work is trying to understand, well, what is the right methodology of sequencing or integrating multiplex data? Is it spatial transcriptomics? Is it actually a multiplex IHC? What are the right methodologies to do this? Those kinds of exploratory assays are not easy to do in the regulated environment of just industry. They have to do it actually in an exploratory way with academic institutions. So I think that's where the opportunities are. They have to be prospective and it has to be partnered.
Okay, well, thank you again, Dr. Gandhi.